So what I'm really going to talk to you about today is drug use from the broader perspective of plant neurotoxins, or how do humans exploit plant neurotoxins. Before I get there, I'm going to set the stage here a little bit, the problem. So this is a chart showing infectious disease mortality and non-infectious disease mortality. And infectious disease mortality is in the red there. We have mortality rate per 100,000 individuals on the y-axis and year across the x-axis there. And what you can see pretty clearly is that red line's dropping. Okay, so infectious disease mortality since the 1900s has declined significantly with a little spike there, the influenza outbreak around 1920. In contrast, that blue line is non-infectious disease mortality. So all causes of mortality not related to infectious diseases, and that has pretty much remained flat. So the question becomes, why haven't non-infectious disease mortality, why haven't those rates dropped as well like we've seen with infectious diseases? Part of this is because we think that germ theory of infectious disease is actually correct. So, there's microorganisms, some of them are pathogenic, and these cause diseases, viruses and bacteria. And we've done a pretty good job of identifying this and developing solutions to deal with these pathogenic microorganisms. But not necessarily with non-infectious disease mortalities. And maybe we have the wrong model for non-infectious disease mortality for a lot of them. So to put drug use in context here, you can see on the bottom right there, about 20% of all the non-infectious disease mortality is related just to tobacco use. And tobacco doesn't make the news as much as it used to, but what is making the news today is opiate abuse in the US. So this is since from 1999 to 2014, and it shows an increase in overdose deaths in the United States during that time. These rates are they're out of 10,000, I believe, so you're going 0 to 2 out of 10,000, and as it's getting redder and redder, that's over 20 out of every 10,000 people. But you could see, this is across the whole United States. There's pretty much not an area that's immune to this opiate epidemic that we've been having. So it's clear that we don't quite have the model for drug use because we have these rates that are increasing for things like opiates, but even tobacco and other drugs, illicit drug use, these are continuing to be a problem, a public health burden. There's two basic mainstream models of drug use. And these are not necessarily wrong models, I'm going to argue, but maybe not complete, the whole complete story. So the first one is the reward model, that nicotine and other drugs contain compounds that make them highly addictive. So they stimulate reward and reinforcement circuitry in the mesolimbic dopamine system in the brain. The other one is the political economic perspective, really having to do with marketing and tobacco companies spending a lot of money, billions of dollars each year, especially now in the developing world, trying to market tobacco and other products. So I'm just going to switch now to thinking about the reward model. Because this reward model is the mainstream model of drugs, the mainstream biological model of drug use. But let's just understand what the mesolimbic dopamine system is, what dopamine is trying to do. So we have reward circuitry in the mesolimbic dopamine system, what we call the MDS, and this regulates and controls behavior by inducing pleasurable effects. It's trying to reinforce behavior. This system it actually evolved to respond to natural rewards. Rewards that would correlate with increased reproductive success. So sex, things like sugar, the idea, sugars and fats, the idea here being that during a hunting-gathering lifestyle, these things were hard to come by, but they are essential foods to take in, so we are highly motivated to seek these substances out. And the compound responding to this is thought to be dopamine. There's dopamine neurons in the ventral tegmental area, projecting to the nucleus accumbens, dorsal striatum, and the prefrontal cortex there. Now, in humans and other animals, there's actually some plant toxins or plant chemicals that stimulate reward and reinforcement circuitry. Mainstream models of drug use in neuropsychiatry, they look at this as an accident. And my colleagues and I have framed this a hijack model. Basically, drugs like nicotine, opiates, these things arguably were not very common in our evolutionary past. And so they're evolutionarily novel. So the fact that they stimulate reward and reinforcement circuitry is a complete mistake, a complete accident, because they're assuming here that these are not natural rewards. And the effects they closely resemble those of natural rewards, so they produce approach and consumatory behavior, positive feelings, and the learning of cues that predict availability. I should also point out that we actually don't know the complete function of dopamine yet. Yes, it is very, very important for reward and reinforcement behavior, but we're also now learning that it also responds to aversive stimuli. And so there are several different models of how dopamine responds to stimuli and reinforces behavior, and we don't quite know for sure which one is the correct model. Okay, so away from the mesolimbic dopamine system and the reward model, getting to just what does drug use look like in populations? Is there consistent trends that we can see cross-nationally? And indeed there are. One of the largest, most consistent trends that we see is age differences. So this is cross-national data from United States, China, South Africa, Ukraine, data from Degenhardt et al., who pumps out a lot of publications on substance use. But we have tobacco on the left and cannabis on the right. And these are data for anybody that's ever used. And then it maps on their age at first use. So what you can see is pretty much nobody uses substances between about under 10 years of age, but we like to say about under 13 years of age. There's no substance use going on here for tobacco, for cannabis. And this trend is consistent across all drug types that we see. So children are basically not using it. And the first time you're ever going to use is somewhere between about 13 and 20 years of age. If you've ever had a cigarette in your life, if you ever smoked cannabis in your life, you were probably around that age when you first had it. That's not all, though. There's also consistent gender differences, and this is kind of a confusing slide, and I'm going to show a clearer slide in a second. But what these depict are what we call odds ratios. What are the odds that a female is going to smoke? What are the odds that a male is going to smoke? And you divide them. Anything under 1, so I'm going to show you this first column here under alcohol. Anything less than 1 means it's a male bias. So we can see in all these red columns, and asterisk means that this is a significant, or what we would expect to be a real difference between men and women. So we can see alcohol, tobacco here, cannabis over there, and cocaine over there. All of them are male biases, and most of them are significant. So there's a significant male bias for all these substances. There's a few where it's like right here, this is just a limited age cohort of 18 to 29-year-olds, and there's a female bias here, but it's non-significant. So the only significant findings, the real effects, gender differences, are male biases. Here's a clearer picture, though. So this plots male smoking prevalence on the X and female smoking prevalence on the Y here. And so anything that's going to have a male bias for smoking is going to be on the right side of that line. And pretty much all of them except for Sweden. Sweden's a little interesting anecdote here. More men than women actually end up using tobacco in Sweden because more men are using snus, a smokeless tobacco. This is just plotting cigarette smoking here. But you can see all the countries are basically to the right, minus Sweden, but we could also kind of put them to the right if we consider the snus that's also being used. There's also a divide here between developed and developing countries. I'll come back to this in a minute. So there's clear sex differences and there's clear age differences cross-nationally in drug use. So what might the neurobiology or model of drug use, what might they say about age and sex differences? Well, on the one hand, they would say that anybody with a functioning reward system would be susceptible to drug use. So, do children have a less functioning reward system than adults? Do women have a less functioning reward system than men do? No, at least the data doesn't suggest that. Among children, their mechanisms are indeed quite functional, and if you look at their fraction of their daily diet just in terms of sugar, a highly rewarding substance, they indeed take a substantial fraction of their diet in terms of sugar. So it's clear that the mesolimbic dopamine system, or the reward system of children, is functioning. It's working. And then women, they also, sex differences in motivation, reinforcement, reward, actually are hypothesized to make them more susceptible to addiction. Maybe not use, but to being dependent and developing addiction. But again, we can look that in the US, women are eating as much sugar as men, at least in terms of a fraction of their total body weight, or the fraction of total calories consumed. So, yeah, reward neural circuitry is presumably or probably the same in both men and women. But globally, as I just showed you, we're getting these very clear gender and age differences in drug use. So what is going on here? Well, we can go to the other main conventional approaches, the political economic approaches. Maybe there's differences in social norms. And this is clear that these do have an effect. So there's lots of data showing that things like gender inequality, economic disparities, all these can influence patterns of use, gender differences and age differences in use. So, we have prescriptions against child smoking. We have prescriptions against pregnant women smoking in our culture. So maybe that explains why some of these people aren't using. There's differences in political and economic power, and also differences in access. So we have, if you're under 18, you can't buy tobacco, a lot of legal restrictions on it. So all these have, in part, some effect on patterns of tobacco use. But then that got us thinking, what about populations that don't really have access to this tobacco marketing and other marketing of drugs, and that actually have high amounts of gender equality? Not gender inequality, but gender equality. So that's, I just asked that question. Now you guys get to read it. But so that's exactly, we have access to these unique populations in the Congo Basin called Congo Basin foragers. And there are many of them. So this is where I conduct my research that I'm going to talk about today is here in this group called the Aka, which are on the border between the Central African Republic, this country up here, and Congo. The Baka are a very closely related group. They're in Cameroon. I'll be probably going there this summer, hopefully. And FA and Mbuti, these are really kind of well-known, popular groups. If you've heard about pygmy populations, these are those pygmy populations. We don't like to call them pygmy populations because it's kind of a derogatory term to just refer to their stature, these short people. And in academics, we refer to them as forest foragers, basically. And then there's all these other groups. But they're small-scale hunting and gathering populations. They are not the type of hunting and gathering populations that probably existed, 150,000 years ago. So it's not like we're going back in time and seeing, what we lived like in the past. But if we want to understand those conditions, this is the best we have. Going to populations like this, where the conditions are starting to approximate how they were in the past. They are highly egalitarian. They're also noted for gender equality. So they do have gender norms. It's not like there's a lack of gender division of labor. There's a gender division of labor. They have some gendered norms. But for example, if a female is going to pick up a hunting spear of a male, she's not going to be ridiculed. She can do that. If a female is going to smoke a cigarette, she could do that. She's not going to be ridiculed for doing that. They also highly value autonomy. So not only is there gender inequality, but and autonomy and individualism. So you are free to kind of be who you are and try out the things that you want to try. And this also has a large effect on parenting. Some people have described their parenting as permissive parenting. Maybe that's not quite the right term, but they don't necessarily punish their kids for not listening to them. And their kids are quite free to experiment around and try all the fruits of the forest, so to speak. Now, tobacco and cannabis use has actually been noted quite a bit among these groups. So they've been written about by a lot of the early ethnographers that went into these areas that were writing about the Efe, the Mbuti, the Aka. But there's really, really, really few detailed studies. My master's thesis advisor, and a good colleague of mine, Dr. Barry Hewlett, where's my mouse here? But he performed probably the first systematic study, but it was just going to the Efe and the Mbuti and going to the Aka and looking at patterns of use, looking at the history, looking at kind of cultural norms and attitudes. So it was a cross-cultural study of cannabis use. Bailey here, he was working among the Efe and the Ituri, and he was looking at tobacco and cannabis use compared to wealth, but that was a really small part of a study that he was doing. And more recently, some Japanese colleagues did a kind of cultural description of tobacco use among the Baka of Cameroon. So now I'm going to talk about kind of neurotoxins and parasites, as my title suggests, but before we do that, what we really wanted to do was test this gender inequality hypothesis. So test the role of gender inequality on gender differences in tobacco and cannabis use among a group of these Congo Basin foragers. But it's not enough just to go in there and say, hey, do you smoke or not smoke, because self-reports of behavior are notoriously not reliable or not 100% reliable. So we also wanted to verify those self-reports with a biomarker. So we had them provide saliva samples, and we took those back to a lab, and we were able to quantify them for the exposure of a nicotine metabolite, what's called cotinine. And then we looked at additional economic and cultural factors that might play a role in gender differences and age differences in tobacco use. Excuse me as I get a sip of water. Again, so these are Aka foragers. One thing about Aka foragers is, in a lot of these Congo Basin foraging populations, is that they have what we might be called interdependent relationships with farming populations. In other words, there's not a day that goes by where they don't eat an agricultural food or food produced on the plantation. So they provide labor to the villagers, the farming villagers, in exchange for plantation goods and Western goods that the villagers might buy at the local market. They trade forest products to the villagers for Western goods and agricultural goods. So they have a very strong interdependent economic trading relationship with these populations. I've already said this, but they're egalitarian, gender equality and autonomy. They have access to and smoke tobacco and cannabis. And they're also, and this is really important, in terms of something that makes them unique and very, very different than the types of populations that we would typically be studied by substance use researchers, is they're what's called a natural fertility population. This means they don't have access and don't use modern contraceptives. In other words, many of the women during their reproductive years are often pregnant or lactating. They're also heavily infected with parasites, and I will revisit why that's important as we continue here. Here is just a picture of some of these substances they are using. Manufactured cigarettes, they actually call them blancs, which is French for white. So they used to be sold in white packaging like this. That's why they call them blancs. They're no longer sold in those types of packaging when I was there 7-8 years ago. The farmers grow this locally grown tobacco here, what they call bangaya, in their gardens, and they'll set it out and dry it and roll it up in these balls and sell it. I don't have a picture, I could have put it there, but it would have been of cannabis, which is something they're also smoking. And then what's really interesting is they have this local indigenous plant that grows in the forest. They take the leaves off of it right here. It's called motunga. The Aka also have it, the Baka also have this plant, and they call it botunga. But they take the leaves off and they roll it in a little leaf, another type of leaf, and they smoke it a lot like they would smoke cigarettes. And it's mildly psychoactive like nicotine is. They also use this plant and a T to kill parasites. So they're using it as a psychoactive plant, like a recreational drug, but they're also using it medicinally to kill parasites. So remember, this is an egalitarian population. They really value gender equality. They're autonomous. They value autonomy. So we predicted, or made a couple of hypotheses, that there would be no gender and age differences in drug use. So the data that I'm going to present here were from 3 different field seasons in 2008, 2010, and 2011. 2008 was just going around, and it was a smaller kind of pilot study, a smaller sample, and it involved only self-reports. In 2010, we have a larger sample of 250 people. Mouse right there. And we have, this is where we collected saliva samples. So nicotine metabolizes, nicotine, for those of you that don't know, is a main psychoactive compound in tobacco. And your body metabolizes that or breaks it down so that it can get rid of it into cotinine. And so you can measure cotinine in people's saliva. And you can, however much cotinine they have, that gives you an index of how much tobacco they were recently smoking. Okay, so what did we find? Well, we found this is from 2008, so this is this is self-report data. 30% or 38% of the, 36% of the women said that they were smokers here, 24, and 95% of the men said that they were smokers. And indeed, this is a very, my arrow's a little off there, but this is a very large sex difference. Even just based on self-report, this is probably one of the largest sex differences documented for a population, even though they're egalitarian. This is the cotinine distribution. So if you have 5 nanograms a milliliter of cotinine in your saliva, that basically indicates that you are a recent smoker. So anything, that's a cutoff. So anything above 5 nanograms a milliliter, you were recently smoking. Anything below it, you are either a non-smoker or smoked long enough ago that we can't detect it. And we have all these dots up here are for men, and all these hashes down here are for women. And we basically have a very clear sex differences. This is all the men right here. Basically, all their values are above 5 nanograms a milliliter. And all the females, except for 2, are below 5 nanograms a milliliter. And 95% of the women, according to this data, and it was less than 5% of the females are smoking. 95% of the men and less than 5% of the females. Okay, so if we if we just plot those values, 2008 values from self-report, 2010 values, self-report, and then here's the cotinine biomarker validated data. You can see that the prevalence of men is right here, this is 95%, and the prevalence for females varies. Either way you cut it, it's a very, very large sex difference or gender difference in drug use or tobacco use. The biomarker validated down here is the largest one with 94% of men using and less than 5% of females using. So we can reject this hypothesis. There is a very large gender difference in smoking prevalence. In fact, the largest that's ever been documented for a population. Okay, so what about age differences? I have to admit that we did not have biomarker validated data for children. It was an IRB issue trying to get IRB approval or institutional review board approval to collect biological samples from children. But we were able to ask those that are smokers, retrospectively, when did you start? These are cultural age categories. So Mona for an Aka is like an infant starting to crawl just to about early childhood before you become an adolescent. Bokala and Gondo here, these are adolescent gender-specific age categories. Bokala is a boy and Gondo is a girl. But you can see that if we just go to totals here, of those that smoke, very few of them said that they were Mona, and most of them said that they were adolescents. Now this is hard to map onto those that chart I showed you earlier, where anybody that's ever going to smoke starts smoking between 13 and 20 years, in part because there's a lot of noise here. I mean, what is this MONA age category is very large, and not everybody's interpretation of MONA or where that cutoff point is exactly the same. So were these 4 people, were they children, like young children? Were they just at that cusp of starting to become kind of adolescents? That's not quite clear. So we need much more fine-grained detail. But then I also went to all the camps, and there's 3 different villages, and about over 1,000 Aka living in all these camps, different camps. And I just went to each camp, asked who lives in this house, what are their ages, what are their genders, do they use tobacco, do they use cannabis? And so this is what we have. Females on the left, males on the right, and cannabis on the top graphs and tobacco on the bottom. And you can see that children are not using either tobacco or cannabis, whether you're male or female. There's a clear large gender difference again, but adolescents aren't using very much unless you're a male here. But again, children are not using. So, and there's a clear smoking prevalences by age here. So we can also reject this hypothesis. So this brings up the question then, what is going on with gender differences? Why do we have, and age differences? Why are there gender and age differences in drug use? So we looked at cultural attitudes, other economics, individual preferences to try to get a clearer picture of what's going on here. A lot of these drugs or neurotoxins, tobacco, cannabis, matunga, these are used by the Aka actually to help out with subsistence activities. Typically male subsistence activities. So they believe that what they'll tell you is that smoking these helps increase strength, warmth, and courage, makes them a better hunter, better worker, and a better dancer. So we're like, okay, is that why people smoke in this population? It's not really. At least that's not what they say. The number one reason why people are smoking is probably something very similar to what people would say here. Desire or hunger, their term for it is njala. They basically probably have a dependency on this. They're craving tobacco or craving nicotine. This is warmth, strength, pleasure was the least mentioned one. These are what we call salient scores. Basically, people list things and you value based on their rank in the list. But this is only mentioned by 3 people. And then, there's not a lot of variation in why men smoking. Essentially, all of them smoke. The only ones who aren't smoking, the men, are older men who basically quit for health reasons. But there's a lot of variation in female smoking. So we asked females, why aren't you smoking? And we could categorize these into 2 broad categories. Personal reasons, so things like aversions to tobacco, do not like it, made them sick, and prescriptive reasons, which is maybe a little bit of a misnomer because, again, we're dealing with an egalitarian population, but there's this gender norm. It is there, it's present that tobacco's for men, not for women, and probably relates to the role of tobacco and these other drugs in subsistence. But so over half of these women are saying prescriptive reasons is the reasons why they're not using it. They've tried tobacco, they don't like it, made them sick. They know other people that use it, but it smells bad and they just don't like it. But that's not the only reason what's going on here too, and this probably also has a large influence on that gendered, tobaccos or smoking's for men, not for women, is that men do not prefer wives who use tobacco, whereas women absolutely prefer males who are using tobacco. So we just asked do you prefer a spouse who uses tobacco or not? And females, 79% of them said yes, 11 or 21% said no, and males, all of them said no, I do not want a spouse who uses tobacco. Well, why? What's going on? The females are saying why they prefer a spouse is that they get strength and work harder. So this is, the reemergence of that kind of cultural model of tobacco and drugs again. I want my spouse to use it because Essentially, they will get stronger, they will work harder, they will bring more resources back. Hey, maybe it's compatible if we both smoke, or maybe there's more tobacco if we both smoke. So this is reasons why they don't. Includes both males and females, but things like my spouse is lazy, they don't obey or listen, they get angry, crazy, fight, not their normal gender role, they speak too much, speak too quickly, don't like smoke, gets promiscuous, etc., etc. Okay, so that suggests that there's a lot of these positive values placed on tobacco in this population. So we were also interested in what kind of negative values are placed on it. Are these substances correlated or associated in the minds of the ACA with negative health effects? So this shows that for Blancs, again, manufactured cigarettes, the locally grown tobacco, and the indigenous plant, that almost everybody has had coughing and chest pain from smoking it, prolonged coughing and chest pain. Some of them have even had vomiting or diarrhea, which might be more indicative of acute toxicity. And then we were especially interested, because remember, this is a natural fertility population where women are often pregnant or lactating. So what are their beliefs about smoking while pregnant or lactating? And almost everybody, 89% of women and 95% of men, said that smoking actually, yes, it is bad for the baby. Do not smoke while pregnant. Aquila is a little bit more of a codified taboo or cultural rule. And it wasn't smoking while pregnant was not necessarily an Aquila, but there was a gender difference, probably in the direction you would expect, with more women than men saying it was an Aquila. It wasn't kind of a taboo to smoke while you're pregnant. And I just like to show these because these are really interesting descriptions. So what happens? Why is it bad to smoke while you're pregnant? Baby can cough inside the womb. Baby can get sick, but unspecified. Can't breathe. Don't know why, but it's bad. Baby dizzy, eyes dizzy, gets diarrhea, turns baby very black, stomach turns, baby dies, etc., etc. So maybe something's going on here with avoidance of these substances to try to maintain the health of a fetus or breastfeeding child. So we might expect to see age differences, especially among women, and we actually do. But you also have to control for whether their mother also smokes. So it seems like smoking status, at least among women, is heritable, whether it has to do with something with biology or whether it has to do with just being influenced from your peer social group, your mother, your friends. There's a clear effect of mother smoking and age here. So this is mother does not smoke on the top and mother smokes on the bottom. And if you are between the ages of 20 to 40, or actually 18 to 40, your odds of your probability of being a smoker if you're females is essentially 0. But if your mother smokes, it's about 50%, about 50 odds. Now, if you're over 40, post-reproductive years, your odds of being a smoker are essentially almost 100% for both if your mother was a smoker or not a smoker. So just quickly on the economy of smoking, could this play a large role in why there's a gender difference in smoking in this population? Now, you have to take these data with a grain of salt, because these are not populations that are really strongly embedded in a market economy where they're getting paid every day with a paycheck. They might infrequently get paid when they work for the villagers, the farmers. More often than not, they're given goods, and some of those goods might be tobacco. But on the rare occasions that they are paid, I asked them how much they're typically paid. There's a clear gender difference. So just to put this in perspective, 500 Central African francs is about $1 in 2008. So men, if they work a full day, are making about 50 cents, and women are making about 25 cents. And then, so this shows, the daily income here, and they're spending about half of that on tobacco. Half of that money that they earn, they say they spend on tobacco, and then they take, of that tobacco that they're purchasing, they share that out with everyone else. So they're sharing, they're buying, half their money on tobacco, and a quarter of their money is just spent on tobacco that they just give away to other people. So just to summarize this culture and economy, we see a very large gender and age difference in tobacco and cannabis use, despite those social and economic disparities being minimized. Now, among adult women, we're arguing that it's a concern for health of a fetus, personal aversions to tobacco, and a concern for attracting husbands that are really shaping their smoking behavior. And among men, we have a really, really extremely high prevalence, despite its high economic cost. They're spending, again, half of their money on tobacco, and 95% of them are smoking. But the smoking is linked to success in subsistence activities and also to mating activities, so retaining and attracting mates. The data on children indicate that there is, again, a gender or an age difference, but we have no biomarker data. I'd like to also point out that most theories on hunter-gatherers' social life, cooperation, focus on sharing. Food sharing, meat sharing. Most studies have not looked at sharing of things like psychoactive substances. But tobacco and other products are actually an ideal good that can be shared. They are highly valuable, they're lightweight, easy to transport, they don't spoil, and so they can be shared by Aka in the village and in the forest. They might also play an important role in the sharing-based social organization of many foraging and other small-scale societies. This has not been part of the theory of hunter-gatherer sharing, but the social importance of tobacco and other substances is mentioned by a lot of ethnographers. Okay, so we've been developing a new model of drug use, what we've called the neurotoxin regulation model, that we think better accounts for patterns of drug use, like in this population and other populations. So what's going on here? Well, terrestrial herbivores eat plants, right? They've been eating plants for about 400 million years. And, plants do want some herbivores to eat them. They produce fruit, they have pollen and flowers, and they're trying to attract herbivores and pollinators. But they don't want all their leaves to be eaten. Their leaves are really, really important for photosynthesis. So over this 400 million years that plants and herbivores have been co-evolving with each other, plants started evolving defensive chemicals. One of a number of defensive traits. Some of them are mechanical, like thorns, but there's a number of different defensive chemicals. And some of them happen to be psychoactive. So auriculin and betel nuts, caffeine. Most of us in here have probably had caffeine today. This is a defensive plant compound, a psychoactive defensive plant compound, nicotine and cotinine. So from this perspective, from the perspective of plant biology and plant-herbivore coevolution, we should be aversive to plant drugs, not rewarded or motivated to seek them out. And this has led us, my colleagues, to develop this reward paradox. Why are we rewarded to seek out something that evolved to deter being consumed in the first place? Turns out that most drugs are indeed plant toxins, plant neurotoxins. They interfere with neurotransmitter synthesis, storage, release, binding, reuptake, receptor activation and function, key enzymes involved in signal transduction, and really importantly, they disrupt all major phases of cortex development. So if you're pregnant and you have a child whose cortex is still developing and you expose that child to neurotoxins, it's disrupting cortex development. So it's very teratogenic. Here is a list of different drugs, world's most popular drugs, the scientific plant name, the toxin that it contains, the neurotransmitter that it mimics, and the receptor that it activates right over here. So, cannabis, known as cannabis sativa, there's also cannabis indica, delta-9 tetrahydrocannabinoids. It mimics anandamide neurotransmitter, and is a receptor agonist for the cannabinoid receptor. So it turns out that plant defensive mechanisms or plant defensive chemicals have had a large role on the physiology of herbivores. So we have a lot of different mechanisms that help us detect and avoid plant defensive compounds. Chemosensors, these are bitter taste receptors, things that taste bitter. They taste bitter because they're potentially toxic. And we actually have bitter taste receptors expressed in the gastrointestinal tract. Airway chemosensors, the function of these is not fully understood yet, but this is new research coming out showing where these bitter taste receptors are expressed additionally in the body. We have detoxification enzymes, so enzymes that detoxify plant drugs and help expel them out of the body. So with nicotine, it's cytochrome P450 enzymes. So we have a whole host of enzymes that metabolize xenobiotics, foreign biotic substances. Cellular membrane carrier proteins that help transport toxins out of the body. And then we have aversive learning mechanisms in the central nervous system, nausea, vomiting, conditioned taste avoidance. All of these play a role in detecting and helping people avoid exposure to plant toxins. Oh, I should also say that many of those defensive proteins are expressed in the brain itself, perhaps underscoring the importance of protecting the brain from neurotoxins. So remember, plant drugs are neurotoxins, and they're what we call teratogenic. They disrupt fetal development. They disrupt all those phases of cortex development. So the cost, what I mean by cost, fitness costs. Costs for survival and reproduction are going to be greatest for children and pregnant women. Children have a neocortex that's still developing. Pregnant women have children, in their wombs who are still developing a neocortex. So exposing yourself as a child or exposing your fetus to these neurotoxins is very, very dangerous. It turns out that we have a bunch of children and females are a little bit better at sensing and clearing toxins as well. So it's not just that we have potentially behavioral aversions to these, which I'll get at in a second, but we have physiological mechanisms that are better at detecting and getting rid of these among those cohorts of the population that are most sensitive. So this shows child toxin defense mechanisms. Children are more sensitive. They have more acute bitter taste sensitivity. They have a larger liver body size ratio. So the larger your liver is to the body size, you can clear out, because liver is where all these metabolic enzymes are at, so it can clear out the body very quickly. And faster xenobiotic clearance rates. So they're much faster at clearing toxins than our adults. And the same thing is true for women, especially pregnant and reproductive aged women. They're more sensitive to bitter compounds, they have a larger liver body size ratio, and they're faster at clearing xenobiotics or drug toxins. So indeed, children are using less than adults. This is kind of looking back retrospectively at the data we've already collected. Children are using less than adults, as we would predict. In natural fertility populations such as the ACA, reproductive aged women use less than post-reproductive aged women, which we showed. And overall, women are using less than men. Now, I'm going to this is just looking back retrospectively at that data. It's not a good test of that hypothesis, but I just want to look again at this figure showing male and female smoking prevalence, and now highlight the difference between developing, which most of them are down here, very low prevalences for women in developing countries. In developed countries are more over here. Still more men smoke, but females have higher rates in developed countries. Part of the variation here might be due to gender inequality, and there's good studies showing that indeed it is due to gender inequality. But my colleagues and I, and this is a colleague that she did her master's thesis on this, she looked at reanalyzed this big data set that was testing the gender inequality hypothesis for drug use. And she took all that data and all these different measures of gender inequality, and then she put total fertility in on it. So total fertility is how many children, per capita. And what she found was that controlling for gender inequality and everything, total fertility, this is females right here, has a very large effect on female smoking prevalence rates across populations. So the higher fertility for women in a population, the less the smoking prevalence in that population. So indeed, it looks like fertility is playing a large role on smoking prevalences for women. And this effect was actually larger than the gender inequality. So gender inequality does matter, but total fertility also matters, and perhaps even more. So I just told everybody why they shouldn't use drugs. But the reality is that people do use drugs, and this is a cross-cultural phenomenon. It's not just unique to us here in Western populations. Well, it could be that under some conditions there might actually be benefits to consuming drugs. And we've been testing one hypothesis in particular, but this isn't the only hypothesis for potential adaptive benefits of being exposed to drugs. So where do we take our insights from? Well, we looked at why animals expose themselves to defensive plant compounds. They do it to self-medicate. So this is a growing area of research called zoopharmacognosy, zoo, animals, basically eating plants to self-medicate. And there's two different versions of this. One is chemotherapeutic. This is not like in the cancer sense. This is in, I am sick today, I have parasites today, and I'm going to treat myself by taking medicine and kill those parasites, right? Or chemoprophylactic. This is, boy, there's a lot of parasites in my environment. I better start consuming something to prevent getting infected with parasites. Now, self-medication, this is clearly humans do this. It's the basis of human traditional herbal medicines. There's good research showing that spices, so in fact, there's a whole field of research called drug food, or food medicines, or the food medicine continuum, of which spices would be included. Sherman and Billing here did a lot of really interesting research showing that the function of spices appears to be to prevent foodborne pathogens. They taste great, but the reason that different cultural cuisines use different spices at different frequencies probably has to do with preventing foodborne pathogens. And there's a bunch of different medicinal foods, as I said, food medicines that have part of foods that might be part of your normal diet, but also have pharmacological effects. Oh, geez, not that button. So it turns out that most recreational drugs contain anthelmintic compounds. Nicotine has been widely used as a dewormer. Anthelmintic just means killing worms or intestinal parasites. We're looking at big macroparasites that live in the gut. Tobacco has been used by traditional populations in the Americas to deworm, as a dewormer. So some populations were using tobacco enemas. It's also been used traditionally to deworm livestock. Auriculin and betel nut. Auriculin and nicotine are very similar. Betel nut, which is used in the Old World, this is anthelmintic. Cannabinoid extracts. These are anthelmintic. Coffee and tea extracts, anthelmintic. Cocaine, a lot of the cocaine is cut with an anthelmintic called levamisole, and piperazine-based stimulants. These are, again, anthelmintics as well. So there's an interesting relationship between psychoactive drugs or recreational drugs, and many of them being combined with anthelmintic compounds. So we tested this hypothesis that maybe drug use is kind of an unconscious form of self-medication against parasites. So this is 2010, 2011. We did this for both tobacco and cannabis. So we did the salivary cotinine, had them spit and provide saliva samples. They provided 3 saliva samples over a week. They also provided 3 urine samples, so we can get at THC acid, tetrahydrocannabinoid acid, which is a metabolite of THC. Tells you how much cannabis you've been smoking. We took stool samples, and they counted up the eggs of these 3 species of worms for a total worm burden score. And we also conducted we call it randomized controlled trial. So, or double-blind randomized controlled trial. Double-blind just meaning I don't know, as a researcher, who's in a treatment group and who's in a control group. We did this only for cotinine, but the treatment group received a deworming pill, basically what's called an anthelmintic albendazole. And after they took that anthelmintic, we went back and reassessed their values of nicotine and worm burden. And then the control group received just a placebo pill, which is like a calcium pill. This, I showed you already the prevalence of tobacco in this population, so this is just to show you the prevalence of cannabis use. 50 nanograms a milliliter right here is the cutoff, so anybody on the left side has THC concentrations low enough that they weren't a recent cannabis user. Anything on the right side is levels above 50 nanograms a milliliter, you were a recent cannabis user. This is only for men, and roughly about 2/3 of the men are smoking cannabis. Remember, keep in mind, this is a population where 95% of the men are smoking tobacco and 2/3 of them are using cannabis. And these concentrations are quite high too. This slide just shows sample characteristics. I'm going to maybe skip over this. So this is looking at the probability of cannabis use by wealth and age. And wealth scores were, again, we basically just had to count up material items they had. How many batteries did they have? How many watches do they have? Do they have a radio? Are they wearing a full outfit of clothes? And so wealth score of 1 is low wealth, wealth score of 3 is high wealth. And you can see that this is probability of smoking cannabis. Those that have low wealth in the solid line have a higher probability of being a cannabis user compared to those with low wealth, or compared to those with high wealth. That's actually a consistent cross-cultural trend that, the way it's put cross-culturally or cross-nationally is that low SES status, low socioeconomic status, is a risk factor for your likelihood of smoking cannabis. I don't think that's the case here. I think that it's, they have to buy their cannabis, and they don't have a lot of material wealth already. So if you're a cannabis user, you're spending money on cannabis, so you're less likely to have money to spend on other things. The other thing here is that your probability of being a smoker is much, much greater if you are younger. And this is another clear, consistent cross-cultural finding that it's typically younger men who are the cannabis users. This is the worm burden, just to show you that indeed they are heavily infected with parasites. There's 98% of men and women were infected, and almost half of them were infected with 2 species, and over a quarter of them were infected with all 3 species of worm. So we actually ran, I'm going to show you 4 hypotheses comparing cotinine or THC concentrations to worm burden scores. The first one here is just a correlational study. This recreational substance use, does it treat current helminth infection? So is looking at cotinine versus helminth burden and THC versus helminth burden. This is cotinine here, and this is a significant effect. There it is right here. So cotinine concentration here, and this is worm burden score. It's not labeled, I apologize for that, but worm burden decreases as cotinine concentration increases. This is actually really similar to what you find in pharmacology, a dose-response curve where low values of a drug don't do much, and then as you increasingly get larger and larger doses, they start to have an effect here. So this is THC. These are some of the control values up here, age and village, but this is worm burden versus THC on the bottom left here. Again, a significant effect that the more THC you're smoking, or I'm sorry, cannabis you're smoking or THC levels you have, the less worm burden you have. So just looking at the values of these recreational drugs compared to worm burden, the more they're smoking, the less worm infection they have. So this is the randomized control trial. Those that are in the treatment group, they should have significantly less salivary cotinine concentrations relative to the control group, because we're removing their parasites. If parasites have anything to do with people's smoking levels, we're expecting those smoking levels to decrease when we remove parasites. And that's what we found. So this is the control group over here, and this is the treatment population over here. Compared relative to the control group who received the placebo pill, the control group got their cotinine concentrations after treatment decreased, controlling for the baseline worm burden scores. So hypothesis 3 that, and again, so I've talked a little bit about xenobiotic metabolizing enzymes and their role in detoxifying the body, clearing out drugs from the body. And with nicotine, it's these cytochrome P450 2A6 alleles, or otherwise there it is, CYP2A6 right here. And there's variation in these genes, right? There's some alleles that, that code for fast metabolizing alleles, some that code for normal metabolizing alleles, and some that code for slow metabolizing alleles. So we kind of hypothesized that those with slow metabolizing alleles would have more of the toxin in their body and therefore less worms. It's not exactly what we found here. This is slow metabolizers, and this is extensive metabolizers. And it's a weird, you have to compare them, but in these middle ages, not at young ages and not at older ages, but in the middle ages here of our sample, those that were extensive metabolizers, those that were slow metabolizers had fewer worm burden relative to the extensive metabolizers. But that effect did not stay true for older ages or younger ages. And it was independent of the amount of toxin in their body. So really it was just being a slow metabolizer predicted less infection. Okay, and then we went back 1 year later, and we said all those people that we treated, what are their reinfection scores? Because we basically removed their parasites. So is smoking having an effect on how they're being reinfected? And this is what we find. So we find that the more people are smoking, so this is year 1, cotinine concentrations, their cotinine concentrations were predicting less reinfection. So the greater you're smoking, the less parasite reinfection you're getting. This is tobacco, and this is cannabis. For both of them, we had this effect. The more THC you're smoking, the less helminth reinfection you have. We kind of interested, well, what's having a stronger effect here? It turns out that cannabis is having a stronger effect. The sample size for these reinfection studies is really small, so we had to remove an intercept here to get a degree of freedom. But in a nutshell, basically, it was THC that has a stronger effect and is more significant on worm burden scores. Okay, so we found support for this pharmacophagy hypothesis in all the tests, with some caveats. So the cross-sectional studies, indeed, the more people are smoking, the less parasites they have. In the RCT study, the treatment group had less parasites relative to the control group after treatment. In the phenotype study, the slow metabolizers had lower worm burden score than normal fast metabolizers, but only within a restricted age range. And then in the longitudinal study, the follow-up study, tobacco and cannabis levels were negatively correlated with reinfection. So what does this all mean? Well, on the one hand, it shows that there's an important relationship between two major public health problems in the developing world. Intestinal helminth infections, which have a burden of disease on par with malaria and tuberculosis in these populations, but as well as substance use. Now, substance use is increasing in the developing world, especially tobacco use. There's been a lot of tobacco marketing, and that's one of the untapped customer bases that the tobacco companies are really trying to market into these low-income countries. In part because they don't have a lot of oversight and regulation, so they're kind of seen as an easy target. Indeed, our results support the hypothesis that drug use is in part a defense against helminth infections. Now, if this is the case, it might suggest that there's some sort of mechanisms, maybe what we could call pharmacophagy mechanism, things like immune signaling pathways. Immunoglobulin E is an antibody that's really important for defense against macroparasites, like the macroparasites we're talking about in this study. And then there's a bunch of immune signaling molecules, cytokines and interleukins, which are important for synthesizing things like IgE. It turns out that there's a lot of these are more and more we're understanding that they do regulate exposure or regulate people's drug use behavior. So for example, interleukin 15 here, right, I don't know what's going on here, but interleukin 15 regulates smoking behavior. This is a really interesting study done by Liu et al. And colleagues. This is part of the larger pro-inflammatory hypothesis of drug use. People are starting to realize, hey, these pro-inflammatory cytokines and immune signaling molecules are important for regulating people's drug use behavior, but they don't know why. I mean, maybe it has something to do with this pharmacophagy mechanism, or hypothesized pharmacophagy mechanism. I should say, though, that the conventional or most widely accepted view on the relationship between drug use and immune function is that drugs compromise the immune system. But if this were the case, maybe there's some novel cessation of prevention methods that we could develop by exploiting these pathways. This also challenges approaches to substance use, conventional mainstream approaches to substance use, and the nature of drug reward more generally. So I talked way back at the beginning of my talk that most people view drug reward as an accident. We have called this the hijack, that these things were not present in evolutionary time, and that they are only present in our populations today, therefore we don't have adaptations to them. And the fact that they stimulate reward circuitry in the brain is just an accident. They, quote unquote, hijack our brain reward circuitry. Well, could it be that drug reward is functional? That under certain context, not all context, but under certain contexts, that there's benefits to exposing yourself to these compounds. And I'm going to end by saying, the reward model, as far as biology goes, is not the complete story. It's not incorrect, but it's not the complete story. We're learning a lot more about dopamine. I think we really need to consider approach and avoidance mechanisms, so toxin avoidance mechanisms, bitter taste receptors, aversive learning, especially in children and reproductive aged women in these natural fertility populations. They are also part of that story, and we need to consider the evolutionary costs and benefits of exposing yourself to these neurotoxins. I'd like to acknowledge all the ACA participants in the study, the Ngandu research assistants. This whole project was funded by Washington State University's Alcohol and Drug Abuse Research Program. And thank you, everyone.